Okay, my name is Perdigao. I'm a research software engineer at the Rosalind Franklin Institute. And I'm here to present a demo of two pieces of software that we have developed, me and my colleagues, Neville as well. It's the software, one of the software is called Okapi-EM and Redlionfish. They were developed to process biological imaging data. Now, the first one, Okapi-EM, is meant to be used to process a type of data called serial-FiB SEM. So it's image. So it's a type of data that is collected from biological samples to get a 3D representation. Okay, it's a bit complex to explain here, but it's, you collect basically many slices of your biological sample, electron microscope image, and then try to reconstruct in 3D. But what this program does is it's got a few tools that help correct for a few problems with that data. One of them is it gives an estimation of the resolution of your images, which is good to feedback on the experiment, people doing the experiments of how the microscopy of the samples is doing. Another one is to correct for misalignment between the slices. So there's an algorithm that does the slice alignment. And the last one is charge artifact suppression. So it's something that appears in a lot of biological imaging images like that, is that you get a problem of charging artifacts. I show an example here, a quick example here of data from that. So this is how our original data looks like. So going from the top to the bottom, as you cut slices through your data, this is yeast cells, and the charging artifacts are in this. You can see that it looks good as you go to the bottom slice, but there's the two problems. One is that too much movement and these charging artifacts. And this, if you apply all both of these tools, you end up with something that looks like this. It's much better corrected. So after that, you can do segmentation and so on. Now the other tool I've got here is more for dealing with light microscopy data. It's called Redlionfish. It basically does Richardson-Lucy deconvolution. It's a common algorithm for people to use it to correct their data for a point spread function. I'm just going to show you a quick example here, if I can. These are both the napari plugin, which is there. That's data. It's a PSF. So if you just look at the data there, it's a 3D data of HeLa cells on a SEM grid that has been cryo-frozen. Oh, sorry. Just ignore that. Yeah, so that's a cell and that's beads. The beads are supposed to look like tiny spots. So, and they don't look like tiny spots because they're blurred by the PSF. But after you do this correction here with the plugin, which is here, I'm not going to run it. I'm just going to show the result, which is here. It looks much sharper. So those beads appear much sharper than the previous ones. You compare that with this. More important is the location of the cell. It's got much more precision, which is important feedback on experimentalists who want to create lamellas and then target for FiB milling and things like that. So yeah, there you go. So hope you like my demo.